Good morning, everyone. My name is John Morris, and I'm very pleased to welcome all of you to the Memory and Aging Project Participants Meeting. Thank you all for coming. A few housekeeping details. If you have an electronic device, please put it on mute or turn it off. The necessary rooms, I believe, are outside the doors and to the left. If I'm wrong, well, look around. And as always, we will, at the end of today's program, have an opportunity for you to ask questions of the speakers. And on your table, you should find index cards and hopefully some writing devices to write your questions down, and we will circulate and pick up the cards from each table and bring them up here to the podium. I understand that some of you, when you came and got your breakfast and your coffee and sat down, found that you had a wet rear end. This has also happened to the speakers, so. But I think we have it all solved. One last comment, just in case, is when it comes time for the speakers, who you're going to really enjoy, to come to the podium, there's a very narrow space for them to push their chair back and stand up. So if they fall into the curtain, we'll give them extra time to get up here. I want to begin. Did I introduce myself? I'm John Morris. I want to give you some explanation about the terms that we use. I know that all of you participate in the Memory and Aging Project, or MAP. That's where you come for your clinical and memory assessments and meet our staff. But the Memory and Aging Project serves three large grants. So the grants come from the National Institutes of Health, specifically the National Institute on Aging. And one grant is actually called the Alzheimer Disease Research Center grant. That grant was first awarded to us in 1985, so this is our 40th anniversary of being a Knight ADRC. There are 36 total ADRCs funded by the National Institutes of Health across the country. We have two other major grants we have many grants, but two other really large grants. The Healthy Aging and Senile Dementia Grant and the Adult Children Study Grant that also, with the ADRC grant, support all of the research that we do. Every 5 years, we have to resubmit our grant application to the National Institutes of Health, and they review the progress that we've made and the progress that we propose to make in the next 5-year budget period, and decide whether we merit being awarded the grant. Our application to go in for another 5-year budget period for the ADRC grant occurred last year, submitted just this week, a year ago. And it proposes that we be funded for another 5 years. That would be years 41 to 45. And the funding would have begun on May 1st of this year and go through April 30th of 2030. The instructions for us to put the grant together said that we had to have attention to our diversity, equity, and inclusion efforts. The total budget was over $22 million for 5 years. The application itself was 937 pages. And as I say, we anticipated that the funding would, for the next 5 years, would begin last month, but to date, and I'll explain this, no funds have been awarded. Now, I want to let you know that the ADRC grant was submitted last year under a new director, and that's Dr. David M. Holtzman, who, along with Krista Mulder and many others, put this major application together. And Dave is here, I don't know where, but I'm going to ask him to stand. There's David Holtzman, the new Alzheimer Disease Research Center Director. Thanks, Dave. So why don't we have our funds? So there have been, apparently, I hear some changes in Washington, D.C. And there's quite an emphasis, as we all know, in reducing government spending. And new directors have been appointed of key agencies, including the Department of Health and Human Services, under which the National Institutes of Health is governed, as well as a new director of the National Institutes of Health. You probably have read that along with other government agencies, there have been staff layoffs and terminations at the National Institutes of Health, with the result that operations there have been very slow, if they progress at all. There's also a different emphasis about how research could go. Less emphasis, really, my opinion, on the quality of the research, but what the emphasis is, more alignment with topics that the new administration would like to address. And even though a year ago we were required to put in our efforts about diversity, equity, and inclusion, now, if those efforts are discussed in a grant, the grant will not be funded. And grants already funded that explore these topics have had their grants terminated. Some of you know about this. Matter of fact, we know that we have certain key words that we cannot put now in our application because the people who are now run these institutions think that they indicate that we're interested in diversity, equity, inclusion. So here are just some of the words that if they're in our grant, they have a filter, they'll check the grant, and if the word is in there, the grant will be rejected. So we no longer can say in our application that we will recruit men and women. Fortunately, we did not plan any research effort in the Gulf of Mexico. And we steer clear of vaccines. But that ADRC grant that we put in last year did have diversity, equity, and inclusion, so we had to go through this past spring and remove all that language. Had to scrub it. And that clean copy was resubmitted to the National Institute of Health or National Institute of Aging a few weeks ago, and we hope it will now be funded. We also put together a large renewal application for our two other program project grants, Healthy Aging, Senile Dementia, Adult Children's Study. That was just submitted last month. That's even a larger grant, about $38 million. And we have our fingers crossed. So what we are doing now with this large grant is looking at the course of Alzheimer's disease across most of the human adult lifespan. The youngest person is 40, the oldest person is 96. And this new grant proposes 4 projects. We want to look at the research results that we have generated in the Knight Alzheimer Disease Research Center and Memory and Aging Project to what we call real-world populations, population living primarily in St. Louis City, as well as a population that comes to memory clinics. And we'll also have projects that explore new biological markers of Alzheimer's disease and assess their ability to predict the onset of Alzheimer's. So even though we're still waiting to see if and when our funding for the next 5 years of the ADRC grant, we are working with the people we know at the National Institute on Aging, and we're hopeful that those funds will be forthcoming. Right now, we are running really largely on gift funds, and we're very fortunate to have that gift fund support, but it won't last for long. All of this is ironic, in a way worrisome, because this is a time in the Alzheimer's disease research enterprise where we stand to make great strides. Well, many have made great strides. Introduction of new treatments and new ways to diagnose a disease. We don't want to relinquish our momentum. Although we can't predict the future, we do hope for the best. So here is the leadership of the different cores and programs within the Knight ADRC, the Memory and Aging Project. Dr. Holtzman is the director. I now am co-director. Dr. Mulder is executive director. In the clinical core, we have myself, Dr. Long, and Nicole Elmore. Biostatistics, Kenji Zhang. Neuropathology core, Rick Perrin. Genetics core, Carlos Cruchaga. The fluid biomarker core, Suzanne Schindler. Imaging core, Tammy Benzinger, Cyrus Raji, Brian Gordon. Biomarker core, Celeste Karch. Research education component, Joy Snyder, Susie Stark. Health disparities and engagement core, we took out equity. Joy Ballsbury. Outreach core, Andre Denny, Jessica Mazursky. Cognitive assessment unit, Jason Hassenstab. And clinical trials unit, Joy Snyder. Some of those individuals are here. I'm going to ask them to stand because they represent the leadership of the entire Alzheimer's Disease Research Center. If you're in the back, step forward so people can see you, Suzanne and Andre. If you stand, don't fall down behind you. Here is the makeup of the Memory and Aging Project, the group that all of you participants interact with on an annual basis. We have, my opinion, I've been around a long time, the best clinical core in the entire ADRC network. And it's because of the individuals shown here. I'm not going to go through each and every one, but we have a fabulous group of nurse clinicians. We have wonderful social workers, you know that very well. A great staff full of coordinators and schedulers. Advanced practice practitioners, nurse practitioner, physician assistant. Physicians, cognitive assessment unit. They are marvelous. And we have 4 new additions to this group, and I'm going to ask them to stand and come forward so they can see you. So Kyle Ogle is a new nurse practitioner. Kyle's stepping forward over here. Elliot Estrada is a new nurse. Elliot, are you here? Yeah, over here, great. Angela Carey is up here, and she is a patient health navigator. And Tom Hammond is an accounting assistant. Where's Tom? Here, he's stepping forward right here. Now, part of the ADRC is really the entire ADRC and Memory and Aging Project is responsible for putting on a program of this size, and I think the team does a fabulous job, but I can't call everybody out for recognition, but believe me, this is entirely a group effort. I do want to thank Howard Palmer and Ron Hawley in the back because they keep our AV needs going and working. Marissa Streitz from the Administration Corps not only does great work, here she is, but she has the unenviable task of putting together my slides. So, Andre Denny is in the back, and she's the head of the Outreach, Recruitment, and Engagement Corps. And probably this would not take place if we didn't have Jennifer Phillips, also from that same corps. Jennifer is in the back waving. So staff, new additions to the staff. Unfortunately for us, we have to say goodbye at the end of this month to two individuals. First is Sushi Sathian. Come on up, Sushi. Come on up, all the way up here, all the way up here. Any of you who have had a lumbar puncture, a spinal tap, know Sushi. Sushi is really remarkable at how she is not only getting everything scheduled, but her support of our participants has been just fantastic. So she is here. Sushi has been with us for over 20 years. And stay up here because we're gonna take a photograph. So stay up here. And then Terry Hosto, our senior social worker. Terry came to us in 1989 from the University of Michigan. So, and Terry, as it says, has really done everything for us. Recruitment, informed consent, supporting everybody, teaching up-and-coming social workers, chairing our ethics committee, and she's a support group facilitator. And unfortunately, we have one other person we had to say goodbye to this year. Some of you will remember another social worker, Becky Fearberg. She retired from the Memory and Aging Project in December of 2023, but this February she passed away. So we all remember Becky with great fondness. One of our missions in the Knight ADRC is to always train the next generation of Alzheimer investigators. And three are here: Maren Gregerson, a neurologist; Michelle Rudman, who's a neuroscientist and a neurologist; and Jonathan Williams, a neurologist. What is it that we're attempting to do in the Knight Alzheimer's Center? We want to do many, many things. One of them, perhaps the major theme, is we want to identify Alzheimer's disease as it begins and before it reaches a point that it causes memory loss, dementia. Some of you know that process, from the time it begins in the brain until it reaches a point that dementia occurs is 20 to 25 years. During that time, it's building in the brain, but the individual is perfectly fine in memory and thinking. Why do we want to identify this stage, this silent stage, asymptomatic stage? One, it will teach us about how the process actually develops and how it causes dementia, but also because this is the time, ultimately, that we want to intervene to prevent dementia. It's going to be very hard to absolutely cure Alzheimer's dementia because the reason people get dementia is that brain process kills brain cells. And unless the treatments in a demented person restore those brain cells, we can't really restore them. But it's much simpler to try to prevent. So that's what we're interested in doing. This is complicated. I don't want to go through everything, but what we see is a brain cell called a neuron that has lots of projections that connect it to other nerve cells. In Alzheimer's disease, there's a clumping of a normal protein in the brain called amyloid beta, the clump is called an amyloid plaque. That's not supposed to be there. So the brain's defense mechanism tries to get rid of the amyloid plaque, but they're not very specific, and all of the things that they try to do to kill the plaque kill the neuron and its processes. So amyloid, we think, is key to start Alzheimer's, but it's certainly not the cause of the dementia, because amyloid by itself will not cause dementia. It's the damage done in trying to get rid of amyloid through the tau protein, which aggregates abnormally, inflammation, TDP-43, vascular factors, many factors go into causing Alzheimer's dementia. This is the way we think of this: a long asymptomatic phase in which amyloid begins the process, but then lots of other things happen to cause brain cell death, neuronal integrity, leading to the symptomatic phase, which is the end stage of this long process. We want to intervene here in this asymptomatic phase to prevent this dementia phase. Well, let's talk about you. There are 1,046 participants in the Memory and Aging Project, including 107 new participants in some pilot studies. I've already mentioned you range in age from 40 to 96 years. 60% of you are women. 18.5% identify as African American. And you, on average, have a college education, 16.2 years on average. You come from 25 states, including California, Washington, South Carolina, Florida, and New Hampshire. And I believe we have a participant here today from Texas, do we? Yep, she's standing up over here, and she's standing up from Texas, came all the way. And the longest active participant is Mr. Ted McClure, 38 years Memory and Aging Project participant. Mr. McClure is here. There he is. We ask a lot of you, of course, the clinical and cognitive or memory assessment, a blood draw every 2 years, brain imaging, MRI and PET every 3 years, and lumbar puncture, spinal tap every 3 years. Can you believe 201 participants have had 3 or more spinal taps, and 62 have had 5 or more? It's amazing. We ask a lot of you. We continue to ask a lot of you. So you do all those procedures where it says MAP studies, but we ask you to do extra studies that look at things such as the eye, the retina, look at falls, brain metabolism, driving, and some of these require extra imaging studies. I know that some of you, it gets very confusing. We do, every 3 years, do MRI and PET scans on you, imaging studies. And you get called, would you be interested in doing another study that has imaging? And you say, I just finished my imaging. Why do I have to do it again? Well, in this case, this is different kind of imaging to address specific additional problems. Some of you know that we're now going to have been assessing your memory and thinking via your smartphone. You get a little signal that you should take a 2-minute test. Several times a year you get asked about this, and this is much more real world because this happens when you're at home or at work or doing something else, not in an artificial testing environment such as we have in the Memory and Aging Project. Dr. Hassenstab and his team have pioneered this, and many people at other institutions are following it. We will be doing the smartphone testing going forward, but there will be a new app that Dr. Hassenstab and his team will roll out for you, and you only now will be tested 1 time a year on this new app instead of twice a year. And we'll be helping you make the switch to the new app. You are also known worldwide. We get many requests for you to participate in research here in other areas of the Washington University research environment, but your information, your images, and your data are requested by investigators all over the world. We had 102 such requests just last year alone. Some of you have been asking, can I learn my own results? And the answer is yes. When we informally asked you about this, 80% of you, over 80% of you said, yes, I definitely would like to learn my results. When we call you up and say, would you like to have them? I've got them right here. Only 60% of you do. Which is fine. Not everyone wants to learn their results. But you have the option. You don't have to, but if you want to, we can do it. So this is called the We Share program. And we have another grant renewal process that's going through that will be submitted this July to return results to all participants who wish to learn them. We have many partners here at the Knight ADRC, the Memory and Aging Project, certainly the Greater Missouri chapter of the Alzheimer Association. I've mentioned an ethics committee, Links Incorporated, the Harvey A. Friedman Center for Aging, the Hope Center for Neurologic Disorders, Barnes Jewish Hospital Foundation. Very strong support from the Chancellor of Washington University, Andrew Martin, the Dean of the School of Medicine, David Perlmutter, and our Neurology Department Chair, Jin-Moo Lee, Advancement in Media Relations. And I want to ask Emily Wozniak, where are you, Emily? Way over there. She helps us get those gift funds that are allowing us to keep going. So you can donate after this to just go over there and give your money to Emily. And many donors and friends, including Joanne Knight. And that's why we are the Knight ADRC. Now there's one. I skipped over one group, and that's our African American Advisory Board. That was formed in the year 2000. Norman R. Say was the inaugural chair of the African American Advisory Board, and through their remarkable efforts, we have increased the representation of people who identify as Black or African American in our research from less than 3% now to over 18%. So this is the 25th anniversary of the African American Advisory Board. I know that several members of the board are here, including the chair, Doug Petty. Doug, where are you? Doug is standing up over here. And I know several others are here as well. Will you stand up, please? Richard King, Sidney White, Barbara Foster. Every year we remember Norman Say with a lecture in his honor, and last year it was presented by Robert Turner to look at brain health and aging research with Black men and former National Football League athletes. I mentioned a new era in Alzheimer's disease, blood tests now, but there's also treatment. The treatment doesn't stop the illness, but it can improve the period of time that people can remain functioning by themselves. There are 2 drugs now that do this. They both target amyloid. The drugs, I didn't put them on here. Don't worry about that. There also are drugs that treat the symptoms of Alzheimer's disease that have been around for a long time, Aricept and Namenda. But the 2 new drugs are called lecanemab and donanemab. And Dr. Snyder is going to talk about them. I put this up there just because it's not easy to take these drugs. They are reimbursed by Medicare, at least partially, but they improve the duration of independent functioning of people who take the drugs by and large. Not everyone, of course, responds to the drugs. There are side effects. They're called amyloid-related imaging abnormalities. So people who take these drugs have to have a brain MRI every few months to see if those imaging abnormalities are developing, at which point the drug can be withheld until the imaging abnormality resolves. So these are the drugs, lecanemab and donanemab. I think they're just the start. We'll get more effective disease-modifying therapies. We'll get combination therapies, and I've already mentioned they may have their best effect when we give them to people who are starting the Alzheimer process in the brain but do not yet have any dementia. Okay, so the Memory and Aging Project staff asks me every day, Dr. Morris, are you retired yet? And the answer is not yet. I'm 77 and a half years of age, and I think once both the grants that I talked about at the beginning are fully funded, that probably would be the time I can start the process. But that's not going to happen for at least a year, so that means I will be here for next year's participants meeting. But when I do retire, what will I do? So I have I have no talent, but I thought maybe I'd take up painting. Then I thought maybe something a little more active. Maybe I'll become a cowboy or an astronaut. But maybe I can combine both art and activity, and this is what I'm going to do. I'm going to become a rock star. But as all of you who are retired, retirement gives us a chance to spend more time with family and grandchildren. So there's my family, and My two grandsons, Jack is 10, Thomas is 7, my two granddaughters, Molly is 3, going on 4, and Grace is 1.5. And so I would look forward to that. Today you are in for a terrific program. Here are today's speakers. They're not necessarily in the order they'll present, but I just want to have you understand that Michael Beloy is very interested in studying genetic risk factors for Alzheimer's. The blood that you donate goes to several purposes, but one of them is to study the DNA, the genes. And so he's going to talk about sex-specific genetic medicine in Alzheimer's disease. Angela Karikari at the end of the table is going to talk about a new study that we're proposing, the COMPASS study, to take all of the knowledge that we've learned in the Memory and Aging Project and translate it to people who are more real world, more representative of the general population. Some of you have had skin biopsies, and you may wonder what that skin biopsy is going to be, how it's going to be used. Dr. Celeste Karch is going to tell you how your skin helps understand Alzheimer's disease, and Dr. Joy Snyder is going to talk about those drugs that I've mentioned. So with that, I am going to step down, and we will welcome, and make sure she doesn't fall down, Celeste Karch. So welcome, Dr. Karch.
I'm a little bit shorter than Dr. Morris. So it's so exciting to be here with all of you today. I'm Celeste Karch, and I also lead the biomarker core, in addition to some of the work that we do in my lab, asking questions about how Alzheimer's disease starts and how we can treat it. And so I'm so excited to be here with you today, because today we have treatments for Alzheimer's disease. I mean, this is the dream. It's incredible. But we know that this is just the beginning. We know that we can keep improving these therapies, make them better. And part of also how we are going to improve these therapies is going to really narrow in on how we can match a specific treatment to the stage of your Alzheimer's disease. But when we're thinking about treatments, it's not one size fits all. And we have lots of examples from this in cancer. And we need to understand which people are going to be most responsive to which drug. So when we think about cancer, we think about some cancers are going to be more responsive to a certain therapy than others. And so we want to get you the best drug for your cancer as soon as possible. And we want to be thinking about how we can really do that as well in the context of Alzheimer's disease. So even though we celebrate every day for being here with treatments for Alzheimer's disease, we know that we have a lot more work to do. And we think to really meaningfully do that work and to really understand how to target the drug to the person is to really understand the people, the cells in your brain, and how they're working well and how they're not working so well. And we do that by collecting little pieces of skin. So we want to study your brain, but we don't want to take any of those precious cells from your brain. You need them. You need them. So instead, only about 20 years ago, there was a discovery that led to the Nobel Prize that showed that you can take a little piece of skin. And so this is a biopsy. Some of you have done it. I've done it, because you gotta know what you're asking others to do. That's just from the sort of back, sort of lower back. We give you a little bit of numbing, so you really just feel a little pressure, a little push. And what we get is a little piece of skin the size of about a pencil eraser. And we can take that skin, we walk it over to the lab, and we put it in a dish, and we chop it up with a razor blade. When we do that, that actually tricks the skin into thinking that it has a cut. And so it gets to work, these cells, special cells, migrate out of that piece of skin in order to repair that injury. Those cells are called fibroblasts, and we can keep those cells. We can grow them in the lab. We can do experiments on them. We can, store them and share them with other researchers that want to ask questions. And these cells become so important because otherwise a lot of the work that we do is to look at sort of how disease occurs in an animal, maybe a mouse, but we're not mice. So we want to really understand how these cells, how your cells, are functioning at the cellular level and how that compares to what we already know about what's going on in the brain with the imaging work that you do, or with changes in certain proteins that we can measure in the CSF, or with genes that are expressed that we know from the blood. And so this becomes incredibly powerful. And I'm going to tell you just a few examples of how we use these cells, but I could talk for days. It's so fun, it's so amazing, and if you ever want to come by the lab and see the cells, it's so cool. I would love to have you. Okay, so one example is actually happening right here at WashU, and this was shown just last year, that we can take these skin cells and we can give them sort of the instructions, kind of like a recipe, to make those skin cells kind of become a cell that looks just like the cells that are in your brain, those neurons that Dr. Morris described. And if you get the conditions right, those cells actually begin to build up these sort of clumped up proteins like amyloid plaques, or that look like these tau tangles that we see in the brain. And so what that means is that now in the lab we can ask, what are the best drugs that are going to clear and dissolve those structures that we know are really central to disease? And we can also ask other questions like, even if there's this kind of clumped up protein that's in the brain, how can we make the cell sort of tolerate that better and resist that cell death that's really likely contributing to the dementia? And so this is really powerful that we can look at lots of drugs really quickly to really get at, what drugs are going to be the best. But beyond looking at those cells themselves, just the skin cells, what we can do is we can give the cells another set of instructions, another set of molecules. And what those molecules do is it erases the memory of what the skin cell or fibroblast. It erases the memory of what it means to be a skin cell. And the cells change in their shape, and they become this cell called an induced pluripotent stem cell. So this is a cell just like in kind of the developing sort of embryo. And this cell, if you give it the proper instructions, can become any cell type in the body, any cell type. And so we know the instructions. We have a pretty good idea of the instructions of how to make it into a cell that looks like those neurons that are in your brain. And even the other cells that are really important in your brain that help those neurons survive and thrive and communicate with each other. Cells that are called microglia and astrocytes. And so we do a lot of that work. And I'm going to give you a couple quick examples of, again, how we can use these cells that have become this kind of cell that can become any cell to understand what's going on in disease. So early on, I loved this study that came out. And what they did is they looked at people who carried these mutations that cause really early forms of Alzheimer's disease. And they found that if they look at the brain cells that come from these people, that some people were responding, or I say people, some cells were responding to a drug and sort of reversing the levels of amyloid beta that gets kind of built up into these plaques better than others. And so that could tell us that those drugs are going to be working better in some people than in others. And so we can get that drug to the people who will respond best, and find alternative drugs that will respond best in those individuals. And so it's really powerful to be able to think about how we can find those drugs, but also match the best drug to the best person quickly and in the lab. The other way that we can use these cells is to think about, our brain is not just one cell that kind of lives by itself. It's really interacting in this dynamic space, right? Our brains are 3D. And so we can grow these cells in 3D. And when we do that, they actually begin to start to look like what we call mini brains. Now, they are not doing any of the amazing things that your brains do. But they are, if we let them grow in culture for as many as 6 months, we can look at them over time and see actually there are changes in those sort of mini brain-like cells that follow a pattern of dysfunction, just like we can see if we go back and look at people who develop Alzheimer's disease and how sort of their CSF changes over time, or how their brain imaging changes over time, or even for those who do ultimately pass from disease, what their brains look like at disease end stage. And so to be able to have something where in the lab we can ask, what events are happening first and then next, and which of our sort of favorite drugs are best acting when we time them at certain stages is going to be really incredible, so that when we come into the clinics, that we're getting you the best drug at the best time to have the most meaningful impact on disease. And, I can talk about this for days, so come find me. I'd love to answer any of your questions. But I just want to thank you all. I think from the very beginning when we first started this, I gave a talk many years ago to you all, and you all were coming up and saying, when can I sign up? And I really appreciate we are recognized at our Knight Alzheimer's Disease Research Center as the biggest collection of these cells. And we're able to not only do incredible work here, which I think WashU is the best in doing the best research, but to share these cells with people all around the world to really get at the causes of disease and how to treat them. And we can only do that because we're all in it together and we're working together. And so I really appreciate you all, and thank you so much.
Okay, next we're going to hear from Michael Beloy. Michael?
Thank you. So, as you can tell probably from the title of my slides, I'm conducting research that the government maybe currently isn't exactly agreeing with. I will mention men and women, but I will...
They want you to speak into the microphone.
Oh, is that better? All right, I'll bring it back. Yeah, so as you can tell from the title of my slides, I am actually doing research that the current government might not be exactly agreeing with. I will mention men and women, this is what we're focusing on, but I will advocate for its importance. But as a starting point, I want to first say that I'm really excited to be here. I've been doing genetics research for Alzheimer's disease now for 7 years. It's the first time I get the opportunity to speak back to the participants that contribute to the very valuable data I get to leverage in my research. And actually, anecdotally, I started this part of my career at Stanford as a postdoc. And the first study I published there that we worked on was to investigate how the Cloto gene can protect individuals from developing Alzheimer's disease. And at that time, we were also investigating how it might reduce the amount of amyloid that develops in the brain. But we only had so much data to look into this question. And I discovered at that time that the Knight ADRC data, which we were already using in our genetic analysis, also had large data sets on amyloid pathology available. That was data that was contributed by Dr. Carlos Kuchaga, that you probably know, that we could access publicly, and it really helped us to shore up our findings and to get a higher impact on our work and validate the cloto protein being involved in Alzheimer's disease. So I thought it was a very nice memory to realize when I came here and to speak to you. So like I mentioned, we study the genetics of Alzheimer's disease, and I think it's really important to do this because what we know from ongoing work in the last 30 years is that Alzheimer's disease has a very large genetic component. So from identical twin studies and other type of genetic studies, we know that about 60 to 80% of Alzheimer's disease can be explained by genetics. So if you have identical twins and one of them develops Alzheimer's disease, there's a 60 to 80% chance that the other twin will also develop Alzheimer's disease. And why is this important? Well, because if we can identify the genes that lead to Alzheimer's disease, that gives us insight into the potential therapies that we can target or glean from this, right? The amyloid therapies essentially come from early discoveries of genes that, like presenilin-1, that we know lead to amyloid plaque formation in the brain. So this is why it's really important to study the genetics of Alzheimer's disease. And many studies have been done on this in the last 20, 30 years, but very few, and especially essentially none at larger scale, have asked how is this differing between men and women. And so that's what we set out to do in my lab. We take these very large-scale genetic data sets, like the Knight ADRC sample that we combine with many other cohorts to try to look at how genetic variants predispose to Alzheimer's disease risk. So what I try to show here in the schematic, you can see there's stage 1, we have the symbols ADGC and ADSP. These are called the Alzheimer's Disease Genetics Consortium set of data sets and the Alzheimer's Disease Sequencing Project set of data sets. And within those data sets are samples from the Knight ADRC that contribute to them. And we consider this our most valuable or clean subset of the data because here we actually have very detailed and strong clinical diagnosis of individuals. We're very certain about our genetic associations in these data sets. And then we combine them with larger data sets from biobanks, like the UK Biobank or the Finnish Health Registry, where the phenotypes aren't as clean, but we have even more individuals, right? But combining all of these data together gives us a very valuable design or a very valuable approach to study genetics of Alzheimer's disease. And so what we did here is conduct these very large-scale genetic association studies in a sex-specific way, and we ended up with over 500,000 female individuals and 500,000 male individuals to run these analyses. And this is essentially the main results that we would get from that. This is a lot to digest, but I'll try to guide you through it. If we focus on the top section of this figure on the left, you can see a series of dots that are being shown across the chromosomes from chromosome 1 to 22, so across the genome. And every individual dot in this graph represents an individual genetic variant that has been measured across the individuals. And the higher up the dot is indicated in the graph, the more significantly it's associated with Alzheimer's disease. And what you can appreciate, hopefully, in there is that there's some towers, right? We call this type of plots Manhattan plots because they reflect the skyscrapers of Manhattan, right? And those skyscrapers is the regions on the genome that are associated with Alzheimer's disease. It's those regions that we want to investigate further to identify the genes that may lead to Alzheimer's disease. And in this case, on the top, we see results for women, and at the bottom, we see the results for the men. And in that type of plot, we call it the Miami plot because it's a reflection of the skyscrapers in the water, and we use that to contrast, in this case, when women versus men. And what you can appreciate here, there's these red dots on the top and blue dots at the bottom. Those are the female or male-specific regions that we're identifying. So in this analysis, we've established 14 regions on the genome that are female-specific and 6 that are male-specific. And what I'm not showing here in the interest of time is all the follow-up work we're also doing there to identify at those regions what are the actual genes that seem to be relevant to Alzheimer's disease. And for all of the regions, essentially, we can identify the potential genes that might be relevant to drug targeting here. Now, what I showed you was chromosomes 1 to 22. So these are the autosomes, right? These are, every man and woman has the same amount of autosomes, the same copies of autosomes. But what is unique between men and women is the X chromosome, right? Men have only one copy and they have a Y chromosome, while women have 2 copies of the X chromosome. And so that means that the X chromosome is uniquely positioned to contribute to sex differences, not just in Alzheimer's disease, but in any form of disease or health, essentially. But the X chromosome, in terms of how to handle those data, is much more challenging than the autosomes because it's inherited differently. For instance, men only inherit X chromosome from their father. From their mother. They cannot inherit it from their father. They have to inherit the Y chromosome from their father. And that leads to many different types of challenges when we process those data, when we ask questions on those data. So what we did is take all those data I showed you before, we even added more data, and we developed a state-of-the-art approach to be able to handle the X chromosome in these genetic association studies. And we conducted the first X chromosome-specific genetic association study of Alzheimer's disease. And it's actually the largest genetic association study of Alzheimer's disease to date, including over 1 million individuals. And so this plot is very similar to what I showed you before. In this case, we're right now combining men and women in this figure. It's again a Manhattan plot, and you can appreciate those little skyscrapers standing out, right? And those are areas on the X chromosome where we are identifying that they are associated with Alzheimer's disease again. And one of them stands out, the tallest tower, which has the name SLC9A7 there. So this tower, we, in our study, investigated in detail, and we actually prioritized that gene as a likely contributor to Alzheimer's disease with a lot of strong evidence for it, displaying a mechanism how it can contribute to how Alzheimer's disease pathology develops in the brain. So we were very excited to be able to do this for the first time and already identify a gene that may be a potential drug target moving forward. And then lastly, what we're doing with these X chromosome analysis is now asking, well, we know the genetic signals now for Alzheimer's disease. How does that link up with other important factors to Alzheimer's disease? Particularly brain integrity is of large interest to us. And in matching up such analysis, we have identified now 21 regions on the X chromosome that may be relevant to Alzheimer's disease. And what we can specifically do with that is start looking at which brain areas are more vulnerable in women compared to men. So that's what I'm trying to show you here on these brain maps. The blue areas that I'm indicating on the left there show areas where women, the volume of the brain area may be more affected and linked to Alzheimer's disease. And on the right map, I'm showing you connectivity between different brain areas. And there we can see there are some male-specific areas that may tie into Alzheimer's disease. So by integrating all of these different types of data, we get a sense of how genetics affects the shape of the brain and the connectivity of the brain and leads to sex differences in Alzheimer's disease. So in conclusion, I would say that studying sex differences in Alzheimer's disease genetics is really starting to pinpoint the first disease mechanisms and genes that are relevant potentially for sex-specific therapies. And with that, I would like to acknowledge and thank all the people that have been involved in my lab, my collaborators in the past and moving forward on these projects, the funding and the resources. But especially I want to thank and acknowledge the collaborators I have been fortunate to work with at WashU. I started here a year and a half ago, and especially the Knight ADRC samples that have been so valuable in the work we do. And thank you all of you for your attention and for your contribution here. Thank you.
Thank you very much. So now I mentioned the COMPASS study of trying to go out into St. Louis City and getting a more general population to participate in research. And we've run a pilot study, and Angela Carey is here to give you an update on that pilot study. Angela?
Hello, can you all hear me all right? Okay, awesome. Okay, I will. My name is Angela Carey, and I'm here to talk about the COMPASS study today. First, I would like to introduce our team. I don't want to put anyone on the spot, but if you'd like to stand, that would be awesome. I'm the COMPASS coordinator. Here's Brooklyn Williams, the COMPASS project manager, Courtney Kavakovich, our nurse practitioner, and Serena Cepeda, our cognitive tester. And the four of us began this journey, that is the Compass Journey, in late February. So the Compass Project stands for the Community Partners Brain Health Study and is a project aimed at exploring brain health, memory, and aging at people who represent St. Louis City. And so the idea behind it is that we're meeting people where they live and work, and there is no expectation for imaging or lumbar puncture in the Compass study. And the findings for Compass will be compared to the findings from the overall MAP and ADRC project. And like Dr. Morris explained earlier, we're looking at people that we like to refer to as the real world, and do the blood test for brain amyloid as developed in the ADRC participants perform as well in the St. Louis population. Eligibility criteria for the Compass project, you'll see right here, is 40 years or older with 13 years of education or less. We ask that Compass participants be English speaking and have an available study partner who can answer questions related to their daily activities, memory, and thinking. And both the participant and the study partner should be available and willing to participate annually for the Compass project. And there is a blood draw component that happens every 2 years. Compass is made possible through IFM and the YFC partnership. So IFM provides primary care to a number of individuals in the St. Louis City, and we're grateful to be partnered with them. They also have a partnership with the Youth and Family Center. The Youth and Family Center provides community service and programming to a number of residents of St. Louis City. And they have a clinic space at the Youth and Family Center that's actually brand new, finished last week. We're really excited about it. It looks really great, super clean. The idea behind our collaboration is that we're able to, again, meet people where they're at. And the Youth and Family Center is located at 818 Cass Avenue. Here you'll see what a Compass visit looks like. So when you arrive to the Youth and Family Center, I do greet you at the very front. And then I take you up to the new clinic space, and there is parking available, both at the Youth and Family Center, and there's also street parking. And what we do within the first hour is go through the consenting and intake process. That takes about roughly 1 hour. And there I'm explaining the details of the study, answering any questions that you might have, and just verifying some demographic information. At that point, the study partner then goes to work with Courtney, who again is our nurse practitioner. And here they're answering questions about the participant. And that takes about 45 minutes to an hour as well, meaning that the study partner finishes up around 10:45 or so. And at that point, they're either free to stay in the waiting room that we have at the clinic space, or they're able to leave if they would like. At that point, the participant is doing some cognitive testing with Serena, which takes about 45 minutes to 1 hour. And to wrap up the visit, the clinical assessment is performed by Courtney. That takes about an hour. Again, the participant is answering questions, and there is a physical exam involved. So in total, you're there from 9 a.m. to about noon, meaning that the visit takes about 3 hours. So we really do appreciate you taking time out of your day. And there are snacks and refreshments provided. Okay, the most exciting part. Here are current Compass pilot study participants. I've seen a handful of you here today, so I'm really glad you could make it. We were able to have 26 participants enrolled, which is also super exciting because the original goal was about 15 to 20 participants. So thank you to the team, Dr. Mulder, Dr. John Morris, for all of your hard work as well. We had 20 women enrolled into the Compass project and 6 men. The average age of a Compass participant is 71.7 years old, with the minimum age of 49 and the max age of 90. On average, Compass participants have an education of 12.4 years, with a minimum of 8 years and a maximum of 15 years. And racially, Compass participants identified as 24 being Black or African American, and 2 of them identifying as white. Future directions for the Compass project, as we are hoping to expand to other community centers and community partnerships this fall, including People's Health Centers and Oak Street Health. And then lastly, Compass was submitted to be a part of the HASD renewal that Dr. Morris detailed earlier, and that happened in late May, and we're hoping to receive funding for the Compass project as soon as May 2026. And I would also like to thank the Co-Equal Registry and their team for their work and helping us out throughout the Compass project. And then here's my contact information, my email, and my office number. Thank you.
So there may be no one in the United States who has more experience and knowledge about these new disease-modifying therapies than Dr. Joy Snyder. So she's here to talk about that. Welcome, Dr. Snyder.
All right, good morning. Thanks, everybody, for coming. I'm the last thing between you and questions and the rest of your day, so I'll try to be pretty quick. I'm going to show you a picture that... Speak right into it, okay. This is a picture Dr. Morris showed you a few minutes ago, and this is a cartoon of the things that we think about in treating Alzheimer's disease. And what's different, I think, about my cartoon is I have little Pac-Man and little X's all over it. And these are the drugs that the approaches we are using to try to treat Alzheimer's disease. And the one we're going to talk about most today are those amyloid plaques, again, that Dr. Morris mentioned. They're the pathological hallmark of Alzheimer's disease, clumps of these proteins folded, a normal protein that's folded abnormally, and it's what Dr. Alzheimer saw in the brain 100 years ago. And it's still something we focus on, not the only target. There are many other ones, but this is the one where we've had the most success. And I've got a little circle around that Pac-Man. That's an antibody. So it's an antibody that's targeted against beta amyloid. And antibodies, as you probably know, are made in your body. They combat disease. But we can also make them in the lab. And when they're made in the lab, we can target them to something specific. So the two new drugs we're going to talk about are both antibodies made in the lab that target beta amyloid. They target a little bit different aspects of it. We may talk about that later, but they target beta amyloid, and they help to clear the amyloid plaques from the brain. So the big news is that amyloid immunotherapy is here. We have it now. It's in the clinic. I'll tell you about that. And really, it's based on years of research, based in part on research that you all participated in, so you were very important in bringing these drugs to the clinic. These are drugs that treat Alzheimer's disease, not all dementia, just Alzheimer's disease very specifically. So we have to use some of those tests, again, that you contributed to, the spinal fluid test, the blood test, the amyloid imaging, all those things you've been doing for the last 40 years are now in the clinic, and we're using them to prove that people have Alzheimer's pathology so we can use these new drugs. So we have to use the drugs. We give these drugs through a vein, intravenously. Patients are treated either every 2 weeks or every 4 weeks, depending on which drug they're on. And they do, as Dr. Morris mentioned, have frequent MRI scans. The other thing that's important is these drugs only work when the disease is mild. So what Dr. Morris told you about detecting the disease when it's mild is critically important. And again, what you did at Memory and Aging, coming in and having your loved ones answer all those questions about your memory, doing all that testing, helped us be much more confident in detecting the disease when it's mild. So that's very important. So this slide shows you the results from the phase 3 study of one of these drugs called lecanemab or Leqembi. These results were published in 2022. You'll notice that on the slide on the left side, it's showing you the amyloid PET scan results. So people who didn't get the drug, the placebo people in blue, their amyloid PET scan stayed the same. They still had a brain full of amyloid. People who got the drug, the amyloid greatly decreased, and some of them had a negative scan by the end of the 18-month study. On the right side is the clinical results, and this is using something called the CDR sum of boxes, which, guess what, was developed here at WashU. So again, you contributed to this as well, and this is widely used as a measure in clinical trials. And the CDR sum of boxes tells us if you're normal, so your CDR is 0. As you get to be more abnormal, it goes up from 0 to 18. What's important to know about these studies is that over the course of this trial, since these people were very mild, their CDR sum of boxes only changed by 1.5. So you see that everybody's CDR sum of box, everybody got worse, which means an increasing sum of boxes perversely, but everybody got worse, but the people on the drug got less worse than the people who weren't on the drug. And it was about by 0.5 sum of boxes, which many people in the field say, well, that's not much, you should have done better. But that's a third of the change that is not happening on average in the people on the drug. So that's why we think these drugs are effective. And I'm just showing you that little gap there. What that means is if we look at the CDR, that means things like there's 6 categories for the CDR. So in memory, when you have a CDR of 0.5, your memory's not as good. You might forget conversations. You might not be quite getting all the details of an event you did a couple of weeks ago, but you're still able to remember most of your appointments, do most of your things, remember what happened. When you get to a CDR1, it's more significant memory loss. So you're becoming repetitive. You're not as good at that. So that's the kind of change we're talking about preventing. This is a study done by our colleague, Dr. Hartz, who you've heard about earlier, heading one of our cores. But what they did was they took the data from memory and aging, from everything you did, and compared it to the changes that were seen in these studies, and figured out that if you were on one of these drugs, you might get between 10 to 18 months longer of being independent, being able to function on your own without needing a lot of help. So that's the kind of impact we think these drugs can have, which all sounds great. Sign me up if I have mild Alzheimer's disease, but as Dr. Morris pointed out, these drugs do have side effects. And the biggest side effects, there are two categories. One is an infusion reaction. When somebody gets the infusion, later that day, the next day, they might kind of feel like they have the flu. They kind of achy, feverish. Usually that's mild. It can happen in up to a third of people, but usually it's mild. Treated with Tylenol, Benadryl, Claritin is okay. The other one, the more concerning one, is, again, what Dr. Morris mentioned, amyloid-related imaging abnormalities, ARIA or ARIA, depending on how you want to say it. And these are things that we see on imaging. So most people do not have symptoms of these, rarely they do, but they show tiny areas of bleeding in the brain, which can actually happen without the drugs, but more concerning areas of swelling in the brain. And these can be large or small. Usually they're pretty small, usually asymptomatic, but we do monitor for them. And as Dr. Morris said, if somebody has one of these, we might stop the medication either permanently or for a little while. Rarely they can be symptomatic. So in 2.5% to 3% of people, they can have symptoms. They can get more confused. They can fall down. They can be dizzy. So in that case, we definitely stop treatment. And rarely these can be fatal. So there were 3 fatalities in the 1,000 people in the first clinical trial. So these are not drugs that we take lightly. So how are we rolling this out in the clinic? So we keep changing the name. This is the current one. Right now I'm calling it the Advanced Alzheimer Therapy Clinic. I'm going to see if that catches on. But this is a clinic that we have for patients that we've seen that we think have mild Alzheimer's disease and might be eligible for the drug. So they come to the clinic and they get a very specialized assessment. And Dr. Morris said, I might be the person most experienced in the country. Actually, it's probably Madeline Paczynski, our PA, because she's been seeing people in this clinic for over a year. Alex Venuto joined her a little later. So Alex and Madeline see patients in this clinic, do the assessments. They're helped by Zach Posey, who's one of our medical assistants, who is a whiz with spreadsheets. So we have a spreadsheet that's on version 2000 now, I think, that tracks all these patients. Leslie Green joined us most recently. She's a nurse who helps out. And then any of you who are in our clinic know Dawn, the amazing nurse who runs the clinic. I get to have the title, but she actually does the work, so it's great. So that's our therapy clinic. So it's staffed by these folks. They see people, talk to them about if they should be on these drugs or not, and then monitor them for the time that they're on the drugs. And this is the really exciting part. So this is where our clinic is the leading academic clinic in the country for treating people with these medications. And this just shows you the number of people we started each month. The orange line is lecanemab. That was the drug approved in 2023. And then donanemab is the newer drug approved in 2024. So we are treating people with these drugs, starting them every month. And we now have over 360 people on these medications. So this is pretty exciting. So we're very excited about having the folks on these drugs. This required a lot of increased effort, both from our physician clinicians as well as from our nurse practitioners, more staff time. People have to have MRIs. We have to follow them. We have to make sure they don't have side effects. We have to answer their questions. None of this is reimbursed. So we're in a clinic. We don't get grant funds. We get funds from people's insurance who see us, and those funds do not cover what we do. So Barnes Hospital has been very helpful and helped us pay for a lot of this staff. So this is great. We're very excited and patting ourselves on the back because we've seen these over 300 people, but guess what? That's a drop in the bucket. There are probably 3,000 to 5,000 people in our area who could be treated with these drugs. And right now, we're the only BJC providers giving them. So we do need to expand. These drugs need to be known better in the clinic, but it requires people who know about them because they do have these side effects. So we've got a lot of education left to do. This is a chart I stole from our nurse practitioners. They developed it, but this is what they tell the patients about these drugs. They compare the drugs. What's the cost of the drug? It can be quite expensive. Medicare generally covers it. Medicare Advantage plans sometimes do, and other insurers are variable, so that's a lot of time we spend is figuring out if your insurance will cover it, how much your copay will be. That's pretty exciting. We look at how much your copay might be, and that's, as many of you know, if you've ever gotten a medical bill, who knows how much things are going to cost. So that's, I think we won't know this for several years, but we're doing our best to try. They slow progression, and you might say, well, I'm going to take the ones that's 33%. Really, the studies were done a little differently. You can't compare them directly, and that's within the margin of error. These are not, our assessment of clinical progression is not super accurate. So for us, these are pretty much equivalent. You might say, well, great, I want the one that's every 4 weeks and not every 2 weeks. That sounds much better. In fact, there's a little difference between side effects, so sometimes we have to talk about that. It depends on your ApoE genotype. You guys have heard about ApoE before. And then the other difference between the drugs is that donanemab was designed to be treated for 18 months, 12 to 18 months till amyloid is cleared and then stopped. Lecanemab is not stopped. You can keep taking the drug. They think it might still have some efficacy even after the plaques are gone. So we still have a lot more to learn. We don't really know which one of these drugs is better right now. We talk to each patient, and this Madeline and Alex spend a lot of time with the patient and their family talking about, what their goals are, what their concerns are, and so people make individual decisions on which drug they want to take. Hopefully in 5 or 10 years, it'll be much clearer, and as Dr. Morris said, we'll have, and I think Celeste mentioned also, we'll have more drugs. So we won't just have one drug that slows progression by 20 or 30%. We'll have a combination of drugs, and maybe we can stop this disease from progressing. So that's where we are with that. We have completed our first year, and Madeline led the charge, along with, I think, Marin's here and some of our other authors on a paper, to report on our experience, because we did have more than other folks. Oh, is Madeline here? Where are you, Madeline? Stand up. Oh, she's over there, way over there. She's standing up. Yeah. So, and we talk at this group, and sometimes we talk about papers that we write, and it's not just like your high school paper where you sit down and write it, for me, the night before it's due. But this took months, and this took a ton of work, and this paper was based on clinical data, which means Madeline and the other collaborators on this paper went through our patient charts with a fine-tooth comb to get the data to where we could present it to our colleagues, have complete confidence in it, because, in the clinic, we're not doing a research study. We're not entering things in a database. We're writing a note, and all neurologists are special. We all write notes differently, so it was a ton of work to put this together. But what we found was very reassuring. So when we do a clinical trial, people have talked about real-world populations. So these folks in the clinic are not like people in a trial. They have other diseases. They're older. They're on different medications. In a clinical trial, people are really, they can't have any other significant diseases. If they do, they have to be very well controlled. So this was a real-world look at this medication. And the side effects were very similar to those reported in the clinical trial. So that was very reassuring, because some people thought it would be much worse. It was a real-world cohort, but it was not diverse. Our clinic largely serves people we only serve people who have insurance, right? So we didn't get uninsured people. We have a largely white population, very little diversity in the population. But we were excited, and our patients mostly had very mild Alzheimer's disease. Some of them had more, not very mild, but mild. And we found the number developing ARIA was pretty much what we expected, about 20%. And very few of those had symptomatic ARIA, only about 6%. So that was very reassuring. We did find infusion reactions did happen, a little more frequently than we thought. For the first time ever, we had to be on call as physicians. We thought there were no dementia emergencies for the last 20 years, but suddenly I had to be on call, and as did Dr. Morris and our other practitioners. Most of the calls went to Madeline and Alex, so that was great, but at night we did take calls. Most of the ARIA we saw was asymptomatic. People didn't know they had it until we saw it on the scan. We did have some people who had symptoms. The striking finding was that the symptoms were much less common in the people with a very mild disease, those people very early in the disease. And people with more severe disease had more symptoms. We sort of had hints of this in some of the clinical trials, but this was a very important observation for us and points out that we really need to identify this disease when it's very mild. That's when these drugs will be most effective and when they'll have the fewest symptoms. So that was pretty exciting. So that was the main finding of this study, was that people with very mild symptoms do the best. So what does this have to do with you? Again, as I mentioned, the reason we can do these trials, test these drugs, is because we now have biomarkers. We have spinal fluid, we have PET, we have blood tests. And the data that you contributed over the last 40 years made these tests possible. One of the best blood tests was developed here at WashU, largely using data that you contributed. So you're in large part responsible for these new drugs. Secondarily, the assessments that we developed enabled to detect mild disease. So that is really important. And again, you contributed to that. So as Dr. Morris mentioned, these are kind of trying times for us from the grant standpoint, but also very exciting because we finally have drugs that can modify the underlying disease. We hope to have more, we hope to press on, and we can only do that with your help. So thank you all, and I think it's time for Q&A. Oh, wait, one more. Oh, you can clap, but one more thing. I almost forgot, as we have a person here who is receiving one of these drugs, and Andre was going to get them a microphone.
Isn't this great? This is my second time coming here, and I find it so helpful. So thank you all very much for these presentations. And I just, it was like I said, I just want to share with you what my experience has been. And for a lot of you here, I know this is going to be very familiar information, okay? It all started with me. I'm 81 years old, a retired clergyman, and I've been with the program for about 2.5 years. I'm receiving Leqembi every 2 weeks. Things started for me a couple of years ago when I started having trouble remembering names. Even people I knew very well, people I had known for many years. And it wasn't like I had forgotten the people, it's just the name wasn't coming, you know, when I met with these people. And I started to get worried about it. So at that time, a church friend of ours was the, the head of the Department of Gerontology at UMSL. So I took him to lunch one day and sat down and talked about it. He told me he didn't think I had a serious problem, but he suggested to me that I get involved in the Memory and Aging Project out of WashU. And I am really grateful for him for that information and very glad that I did so. Because, well, you know, a lot of you know what it's all about. You know, I went through all the testings, the MRI, the PET scans, the CAT scans, the blood work, this, that, and all the other thing, you know. And the second time I went through it, a PET scan detected that I was in early stages of Alzheimer's. And as they've been saying, it's important that you catch it early. And so there again, I'm really glad I've been involved with Memory and Aging Project. I also started having problems with words. Words are a big thing with me. You know, I was a preacher for a long time, giving sermons every week, a lot of times with no manuscript or anything. And this was fine, but all of a sudden I started to have issues not finding a word I was looking for. And one of the things I like to do is I also like to sing. So I frequently do open mics with either my guitar or my banjo. And I know hundreds of songs, have known them for many years. And I'd find myself in the middle of doing a song and all of a sudden, words aren't there. Where are they? You know? And they're still there somewhere. The problem is access, right? As you've been hearing in these reports, there's these amyloid plaques are interrupting the brain waves in there. So then I started Leqembi, some of you may be wondering, how is this going to affect me? How am I going to respond to the drugs? Well, I'll tell you what my experience has been. The first time it was pretty rough on me, and I'm glad I had my wife with me the first time I went to get an infusion. Because I was kind of wiped out afterwards. I was feeling out of sorts. The whole next day I had zero energy. I spent the whole day on a recliner. We called in and they said, well, you know, a little Benadryl and Tylenol will help, and they helped some. By the next day, I was almost back to normal. I was a lot better, had a lot more energy. The day after that, no more side effects whatsoever. The next time I went in for my second infusion, I had some of the same thing, but less than half as bad. It was much more manageable. Now, it's a piece of cake, okay? It's a piece of cake. I almost look forward to it. I'll tell you what. The worst part of the whole thing is dealing with traffic on Kings Highway, trying to get to Barnes Hospital. All the construction and everything, it's awful. But, you know, once you get there, things start to get good. Because, for instance, you know, you're not going to the regular Barnes building. You're going to the special, what do they call it, the advanced medicine, Center for Advanced Medicine, which means you get to park in the north parking lot. You do not have to go to the south parking lot, which you want to avoid at all costs, believe me. But so you get in the north parking lot, and you walk across the bridge on the third level floor there. You take the elevator up to the 10th floor. You follow the signs for the outpatient infusion center. You go in there, and there's a friendly lady at the desk who asks you, what is your name and your birth date? Now, assuming that you remember that, and that you're there on the right day, and reasonably close to the right time, she'll have you take a seat. And they're very good about this. I've never had to wait more than 5 minutes. They're very well organized. The last time I was there, I went to the seat and I started to sit down, and the nurse was calling me in, so I didn't get to sit down. Am I going on too long?
You are doing such a great job, and I think we could listen for a long time, but I know that they also want to get to the questions, and
Okay.
so.
Okay, this is for Dr. Beloy. Does hormone imbalance explain the increased risk of Alzheimer's disease for women?
Just checking this work. Okay. Yeah, so that's a very good question. A lot of research is going on.
By the way, I'm sorry, Michael. When we say that's a very good question, it means we don't know the answer. But go ahead and try.
It's quite true. I was about to get there. I think there's a lot of good research going on into that. We're actually also doing some of that ourselves in the lab. We know already for a long time that estrogen decline and the treatment that follows, the timing of that treatment, seems to link to brain health and potentially also to Alzheimer's disease risk. What we're observing more recently, I think, which is very interesting, is that there is a link, for instance, between testosterone levels, even in women, that can protect against Alzheimer's disease. So there is ongoing research here. We're convinced that the level of hormones or the differences that occur there across the lifespan are relevant in both sexes, but there's definitely still a lot to learn. So I don't want to give a too conclusive answer on that.
Great. Here's a question, I think, for me. What will happen if the grants are not funded? I don't even want to think about it. I think the grants will be funded. I'm optimistic. If they're not funded, then we are in trouble. We won't have the scope of what we do available to us. I don't think we will go away, but we'll have to constrict what we do. And that's what I think I tried to say earlier. It is absolutely the wrong time to constrict. We need to expand. But I am hopeful for the future. You've seen such a fantastic team. This research is so important, and I think ultimately people will see the value of continuing to fund Alzheimer research, not only here at our place, but across the country. This is a difficult question. At what age should people be tested for biomarkers? Dr. Snyder?
Great. Yes, okay. Great question, as Dr. Morris said. At this point, biomarker testing is only for people who have symptoms. So we only have medications approved for people with symptoms. So the answer is there's no specific age yet, but if you have memory symptoms, you should see your doctor and get assessed. And we shouldn't really even test everybody with memory symptoms because there are many other causes for memory symptoms besides Alzheimer's disease. If we test everybody and we say you've got a brain full of amyloid, but nothing we can do about it because you don't have symptoms of Alzheimer's disease, that's not so great. We are exploring that through the We Share project here in our program, so we're learning more about that. So for right now, no specific age. In about 5 years, in 2028, we will have the results of the first prevention study using these drugs to see if we can prevent Alzheimer's disease. And if that study is positive, then yes, maybe we'll start testing people at age 50, at age 60. Really, the way to find this out is through all of you continuing to give us your blood every year and continuing to do all these studies so we can find out at what age would be the best, what age could we detect these changes, and then hopefully by then we'll know what age we should treat people. So someday, in addition to your colonoscopy, mammogram, prostate exam, you could hopefully also have a blood test for Alzheimer's disease.
You are such great participants and so knowledgeable and so really inquiring. A question here, what about body composition, visceral fat as a causative factor for Alzheimer's? Are we doing anything? In fact, the Knight ADRC is doing something. I mentioned in the imaging core, Dr. Benzinger is the core leader, but Dr. Cyrus Raji is the co-core leader, and he is doing exactly that. And he does find that more visceral fat, belly fat, is a risk for Alzheimer's disease. So those studies are underway. Dr. Snyder, I'm going to come back to you because this is always an important question, and it has to do with what really is the difference between dementia and Alzheimer's? If you have dementia, how do you know you have Alzheimer's?
Oh, that's a great question. It's our probably most frequently asked question in the clinic. So dementia describes the symptoms, memory loss, trouble with judgment, trouble doing things you used to do. Alzheimer's disease is one specific cause of that memory problem. And in people over 65, it causes 70 to 80% of the memory problems. So sometimes people, the two do have a lot of overlap. But Alzheimer's disease refers to the specific brain pathology that causes that problem. And as Dr. Morris has taught us over many years, Alzheimer's disease starts before the memory problem starts. So it's a long progression. But dementia is the symptoms and Alzheimer's disease is the cause. Other causes could be strokes, dementia with Lewy bodies, frontotemporal dementias, many other things.
All right. Will results of PET scans and lumbar puncture be shared with each participant? So our goal is to ask each participant if they wish to learn their PET scan and lumbar puncture results. We have focused right now on the PET scan results, but the ultimate aim is to allow people to have all of their results, including the blood test results. But as I said, not everyone, when given that opportunity, chooses to do that. And that's just a personal decision, whether you wish to know that or not. Back to Dr. Snyder, what happens when the infusions stop?
Great question. So there are several reasons the infusions might stop. You might have a side effect. You might have bad infusion reactions like we heard about that just keep going on. Usually they don't, but they can. You might have some of those amyloid-related imaging abnormalities. Or you might get, for donanemab, you might get to the end of the 12 or 18 months and we find out you've cleared amyloid, so we would stop the infusion. We can also stop them when the disease becomes too severe, when we know the drug would no longer be effective, but you'd still be at risk of side effects. And after they stop, we continue to monitor people, but we don't, at this point, have other drugs to offer. We do offer folks to come join us in the clinical trials unit, test new medications. Our goal someday is to have many different options of drugs. So we compare where our field right now is to where multiple sclerosis was 20 years ago. They got their first disease-modifying drug. It was very exciting. Not everybody could take it. Now they have, I don't know, I keep having to study this when I reboard. They have 10 or 20 medications that you can use. So hopefully someday we'll be that way in the Alzheimer field, but we're not quite there yet. As Dr. Morris said, it's time for us to expand our efforts and get those new medications.
Angela, will the COMPASS study remain in St. Louis City, or will it expand to include St. Louis County, Illinois, et cetera, et cetera?
Great question. I'm actually not sure what the plans are as far as expansion. Maybe Dr. Morris can answer to that.
So the COMPASS study is designed to try to find people who are more like the general population. The general population of St. Louis City differs from that of St. Louis County or St. Charles County, but we have already a fair proportion of people from those counties who are Memory and Aging Project participants. So right now we're focusing on the city. There are other people in Missouri and Illinois who are unrepresented. Those are people living in rural areas, and I think we do want to expand to rural areas. But right now, we're trying to shore up the population groups that we do not now include in the Memory and Aging Project. So right now, we're focused on the city. A question said that they heard somebody from WashU talking about the new therapies for Alzheimer's disease, and someone made the statement, I think, that Alzheimer's disease will be cured in the next 4 years. I told you I'm pretty old, and I was here when the first drug ever approved by the Food and Drug Administration for the treatment of Alzheimer's disease came out. Matter of fact, we were a study site in that trial. The drug is something you probably haven't heard of. It is Tacrin, or the brand name was Cognex. It was a drug that was approved in 1992. You had to take the pill 4 times a day, so hard, and it caused liver toxicity. So the next drug to come along was called donepezil or Aricept. That was a once-a-day drug, very few side effects, and Tacrin went away because it was so hard. But I remember being interviewed, first drug ever for Alzheimer's disease, 1992, Tacrin, and the reporters wanted to know, well, what does this mean for curing Alzheimer's disease? And back in 1992, I was younger. And I said, well, pretty confidently, I said, well, I think in the next 5 years we'll have a cure for Alzheimer's. So I've learned that this is a very difficult disease. We have made great strides and we're on the road, but it's going to be a while before we cure Alzheimer's disease. I'm looking at prevention, but it's going to be a while before we get there too. And by the way, if we can find that one of these drugs or new drugs to come along do prevent Alzheimer's dementia, then the question about when should I get a biomarker test becomes very different. I mean, I would say that, oh shoot, I was looking to see if any of you are Medicare age. I don't think any of you are. There's something called an annual wellness visit that Medicare supports, where you get checked for diabetes and hypercholesterol, and so I can see adding an Alzheimer biomarker test. And if it's positive, if we have effective prevention, people could go to that. Now, again, not in the next 4 years. It's going to be a while. Another question came up about suicide. Believe it or not, we did a study on this, and the suicide rate of people with Alzheimer's disease is really not higher than people without Alzheimer's disease. I'll tell you another anecdote, and I think it'll explain why. I had one of my patients, a person who was an American citizen, but also had come from Switzerland and retained her Swiss citizenship. I don't know if you know, in Switzerland, you can choose to end your life if you have a terrible disease. And she came into my office the first time and said, if you tell me I have Alzheimer's disease, I'm moving back to Switzerland and I'm going to end my life. And I said, well, let's cross that road when we get there. And we followed her to make sure that what she was concerned about, in fact, was true. It turned out to be Alzheimer's dementia. But by the time it was declared, she no longer remembered going to Switzerland. I mean, just very, very difficult. So I think none of us want to have Alzheimer's, right? And some of us might consider going to a country that allows that. But I think the Alzheimer patient doesn't make that decision. Does that make sense? All right. More really great questions that we can't answer. Angela, this is a good one for you too, and you might bounce it back to me. What about increasing participation from the Latino community?
Okay, I think that is something that we would love to do as a team, the Compass team, but currently we are constricted by the demographics that are represented by St. Louis City. I think that we in the future would like to do more to get Latino participants. Dr. Morris?
Yeah, Angela is absolutely correct. We are open to Latinos. The demographics of the greater metropolitan area of St. Louis are something like 17 to 18% African American or black, about 4% Hispanic, about 3% Asian. Now, people have said, and I think there's some truth to this, probably more than 4% Hispanic, because some don't want to say their ethnicity for fear of discrimination or now deportation, right? But if it's only 4%, we would have to have a very large representation from that 4% to come into the study in numbers sufficient enough to make the analyses worthwhile. If we only have 2 or 3 Hispanics, it's just not enough to make any statistical conclusions from that. But are we open to Hispanics? Absolutely. Dr. Karch, how do I donate skin? What do I do?
So that's a great question that we do have an answer for. So you can do that at your regular visit. And you might get a phone call as you're scheduling, and you might be asked if you'd be interested. And so say yes, please.
Are the 2 new drugs effective for everyone? And rather than go to Dr. Snyder, sir, someone wanted to know, we had to stop your report of your experience with Leqembi. Do you think, and you don't have to answer, but do you think it's benefited you?
Here's what I can say.
Is there a microphone? Okay, can you hear? Okay, sorry, go ahead. Please.
Here's what I can say, I'm not getting worse.
You talked about your first infusion and getting side effects. Any side effects now?
Zero. The second one was less than half as bad. Now I go there and I leave and it's like nothing happened.
Dr. Beloy, increased incidence of Alzheimer's in women or men?
So we know that when we look at the general population, about 2/3 of individuals that develop Alzheimer's disease are women. But we know also that's because women tend to live longer than men, right? So if you get to older age, you have a higher chance of developing Alzheimer's disease. But what we also see is that even at these older ages, there is evidence that at that time, still more women are developing Alzheimer's disease than men. So if you try to account for the fact that they live longer. So at the end of the day, it does look that Alzheimer's disease is a bit more prominent in women compared to men.
Here's a very important question. Do diet and exercise make a difference? So we heard Dr. Beloy say that the risk for Alzheimer's disease, about 60%, maybe as much as 80%, is in our genes, but maybe 20 to 40% are other factors. And there is increasing evidence that what we call lifestyle factors play a big role. So you've all heard about things like the Mediterranean diet, right? And it's true that people who take the Mediterranean, have the Mediterranean diet seem to have less risk of Alzheimer's disease. Same for physical activity. Same for, you've heard about this, right? Keeping the brain active. These are all things that may help, and we, Dr. Snyder and I, in our clinics, recommend them. They certainly won't hurt, and they may help reduce risk. But we also have to factor in something else when we talk about people who do Sudoku, and maybe they have less risk of Alzheimer's. Maybe they picked up Sudoku because they had a bigger, better brain to begin with. And it's not the Sudoku that protects them, it's the pre-existing thing. So that's what we have to sort out. But you do recommend it? Yeah, good, okay. We do recommend it. It won't hurt and it may help. Okay, good question. Can imaging be done in participants with a pacemaker? So it's a little complicated. The metal in pacemakers can be a contraindication to brain MRI. Brain MRI is typically done before a PET scan, an amyloid PET scan or a tau PET scan, because it helps. The MRI is much more informative about regions of the brain that are involved than the PET scan. So it tells us if the amyloid is in a certain region of the brain or not. But it is possible that some pacemakers permit MRI. That's one. You'd have to get the information from your cardiologist, and our radiology group would review it. And it's also possible to get the PET scan without an MRI. It's not quite as informative as having the MRI, but it's possible to do. So if anyone is concerned about that, let us know and we can investigate. Okay, I've had a number of questions here that I don't want to be political, but about the concerns about funding, and I appreciate those concerns very much. But here's a proposal. Instead of Alzheimer's Disease Research Center, why not change the name of our grant to MAGA? MAGA stands for Make Alzheimer's Go Away. There's a question about aluminum, mercury, arsenic. Joy, you want to handle that one?
No. No, it's aluminum. Yeah, there was a lot of press several years ago about aluminum, and I think everybody except me threw out their Calphalon cookware. But, you know, none of those things are good for you in excess, but we don't think they're the cause of most Alzheimer's disease. Say what? Plastic? Oh, plastic. Yeah, plastic isn't, don't eat that either. It's not on the Mediterranean diet. Yeah, none of those things are good for you in excess. We don't think they cause Alzheimer's disease. They are certainly, you know, areas of study. Metal chelation gets exciting every now and then, and some people go pay to have that done. We don't think, unless you have a specific disorder of metals, that it's a good thing to do that. So Mediterranean diet, don't eat plastic.
A number of you have questions about exposures to things, not only heavy metals, but mold and other pathogens. We are doing a study of particulate matter in the atmosphere because, not plastics per se, but it certainly seems that areas of air pollution have an increased risk of Alzheimer's, so we are studying that. What markers are the Alzheimer blood tests checking for? Very good question. Dr. Schindler? Left? All right, I will tell you that they evaluate amyloid and tau, the two main Alzheimer proteins. Are you still looking for research participants? Absolutely. Who should an interested participant contact? Email or website? Andre Denny is our recruitment corps leader, and she's going to tell us.
First of all, if you're here and not already in the research, we have back of the table in that back corner some cards that you could fill out, and then someone would contact you. You can also go to our website, and there's a place for you interested in participating in research, and send your information to us that way, and then one member of our team will call and talk to you specifically about what opportunities might be a good match for you.
What is our website?
Knight ADRC.
knightadrc.worcester.edu. Is that true?
It's on our materials too. I'm confirming.
That's kind of embarrassing that I don't know it. Dr. Karch, you were so nice to thank the participants about all that they've done, and you're so excited about the information that these cells can teach us, and you invited people to talk to you. So here, the question is, when is the tour to come to the lab?
I feel like now we need to schedule, Mondays with your cells. You can email me. I can tell you my email. It's Karch, Karchc, @worcester.edu. You can go to the Knight ADRC website and contact. You can call the Memory Aging Project, and they will get in contact with me, and I'll reach out, and we can schedule a time that works for you to come to the lab. I won't be able to know if they're your cells or not. They'll be cell F12455.8, and I will show you those cells and answer any questions that you all have.
Pretty nice, huh? Thank you, Celeste. Are there any treatments for tau? Yes. Just like there are antibodies to amyloid, there are antibodies to tau, and they are starting to be used in combination with amyloid antibodies. So the first experimental studies of combination of drugs has begun. And I can see if the tau antibodies boost the effect of the amyloid antibodies, we can add an anti-inflammatory drug or some other kind of target, other drug that targets another possible factor, just like chemotherapy and cancer. Different targets, different drugs. So I can see that coming. Well, there are so many questions. Again, I applaud you for your interest and for your insight and for your curiosity. We are at time. I think that Drs. Snyder, Karch, Beloy, and Angela Carey once again did a fabulous job. And we'll be willing to answer any questions you may have if you want to come up and address them to you. And on behalf of all of us at the Knight ADRC and the Memory and Aging Project, thank you all for coming, and thank you for your participation. We'll see you next year.